Here are 4 high-yield USMLE-style questions focused on flow cytometry, its applications, and the interpretation of results. Question 1. A 35-year-old woman presents with fever, fatigue, and swollen lymph nodes. A flow cytometry analysis of her peripheral blood shows an abnormal population of cells in the right upper quadrant of the scatter plot, indicating that these cells are positive for both CD8 and CD3. Which of the following is the most likely diagnosis? A. Acute lymphoblastic leukemia ALL. B. Chronic lymphocytic leukemia. C. Normal T lymphocytes. D. Hodgkin lymphoma. E. Non-Hodgkin lymphoma. Normal T lymphocytes. Explanation. In flow cytometry, the right upper quadrant of the scatter plot indicates cells that are positive for both CD8, a T cell marker, and CD3, a marker of all T cells. This pattern is typical for normal CD8 plus T lymphocytes, as these cells normally express both CD8 and CD3. The presence of these cells in the right upper quadrant does not suggest leukemia or lymphoma. Question 2. A patient with HIV undergoes a flow cytometry test to assess the number of CD4 plus T cells. The CD4 plus count is found to be significantly reduced. What is the primary use of flow cytometry in this patient? A. To identify the presence of HIV RNA. B. To quantify CD4 plus T cell count in the blood. C. To detect the presence of HIV antibodies. D. To identify specific mutations in the HIV genome. E. To measure HIV viral load. B. To quantify CD4 plus T cell count in the blood. Explanation. Flow cytometry is widely used in the management of HIV to quantify CD4 plus T cells, which are the target cells for the virus. The STAT CD4 plus count is a key marker of immune function in HIV-infected individuals, and its reduction signifies immunosuppression. This information helps guide the management and monitoring of HIV infection. Question 3. In a flow cytometry experiment, a patient's cells are found to be negative for CD8 but positive for CD3. Which of the following is the most likely explanation for these findings? A. Normal CD4 plus T lymphocytes. B. Cytotoxic T cells. C. Helper T cells. D. B cells. E. Plasma cells. Answer. A. Normal CD4 plus T lymphocytes. Explanation. The pattern where cells are positive for CD3 but negative for CD8 suggests that these cells are CD4 plus T lymphocytes, which express CD3 but not CD8. CD4 plus T cells are helper T cells, which are essential for immune response modulation. In contrast, CD8 plus T cells are cytotoxic T cells and express CD3 and CD8. Question 4. A flow cytometry analysis is performed on a patient with suspected paroxysmal nocturnal hemoglobinuria, PNH. The analysis reveals a lack of expression of CD55 and CD59 on the patient's red blood cells. Which of the following is the most likely underlying pathophysiology of this finding? A. Deficiency of complement regulatory proteins due to a defect in the PIGA gene. B. Autoimmune destruction of red blood cells. C. Increased expression of surface antigens due to viral infection. D. Defective erythropoiesis in the bone marrow. E. Increased production of red blood cells in response to anemia. Answer A. Deficiency of complement regulatory proteins due to a defect in the PIGA gene. Explanation. Paroxysmal nocturnal hemoglobinuria, PNH, is a rare disorder caused by a mutation in the PIGA gene, leading to deficiency of complement regulatory proteins such as CD55 and CD59 on the surface of red blood cells. This results in unregulated complement activation and hemolysis of red blood cells, which can be detected via flow cytometry. These questions